All right, hi AP Bio of the 2019-2020 school year. Hope you are enjoying your sort of snow day. It's January 15th, 2020, and we need to take care of the cellular respiration graphic organizer. So this is just a couple of notes that I have placed on a separate paper. You could put this in your lab notebook, you could put it on the back of your graphic organizer, but this is where I'm going to start. If you just want to watch this lecture real quick and then fill in your notes later, great. Or if you want to have your notes filled out and then follow along as I go through them, the images of these notes are now posted in the PowerPoints and lecture notes folder on Schoology. We're in Unit 4, and this is Cellular Respiration, Chapter 9. Everything that I'm making sure we cover, it's all here on our unit standards. This is kind of like the meat of cellular respiration stuff, and this is some of those leftover locations within the mitochondria that we had from our cell unit. Keep in mind, exclusion statement, you aren't going to need to know specific steps, names of enzymes, and intermediate pathways of these processes. Memorization of the steps of glycolysis, Krebs cycle, and the structures of molecules and enzymes are not important other than ATP synthase. So, let's remind ourselves that all multicellular eukaryotic organisms are going to do this process. Even single-celled eukaryotes like our protists. So animals, plants, bugs, fish, insects, like everything is doing this. They all need energy to be alive, to sustain life, and they're doing this process in all of their cells all the time. So, in order to have the energy to survive, live, like I said, we're getting that energy, and our source is the chemical energy stored in our food. So if you're a plant or an autotroph, we learn that you make that food doing photosynthesis. You don't go out and get a cheeseburger. You don't have the food come to you. You're using the sunlight energy, harnessing it, capturing that energy, and putting into biological molecules that then as a plant you will eat in cellular respiration and basically extract that energy for making ATP to do all of these other things. So everybody else is a heterotroph if you're not a photosynthesizer, so you have to be consuming some sort of food to harness the energy, and that's the process of cellular respiration. So where are we? In the mitochondria of all eukaryotes. So my mitochondria, I like to make them orange or purple because then I know it's not a chloroplast. Remember, it is a wrinkly jelly bean shape. So this is my gigantic jelly bean, wrinkles on the inside. One mitochondria here. There are a couple of structures that you do need to know. So let me zoom in. Okay, we have our outer membrane, outer mitochondrial membrane, the wrinkly inner mitochondrial membrane. The space between those two, we call it the intermembrane space. And the very inside of the mitochondria, inside the inner membrane, is the matrix. It's kind of like the soup of the mitochondria, but there's also soupiness here. We're going to be pumping things back and forth, and those are our transmembrane proteins. That's going to be an electron transport chain and ATP synthase, again, getting things in and out of from the matrix to the intermembrane space, matrix, intermembrane space, back and forth. But the mitochondria has that full outer membrane to basically isolate and house all of these reactions in just the mitochondria. What if it doesn't have a mitochondria, you ask? Well, if you don't have a mitochondria, you are a prokaryote, you're a bacteria, and you are a single-celled organism with no nucleus. So they have similar reactions that are occurring. Here's a cell wall around the outside of an E. coli, and then the plasma membrane inside. The reactions are just occurring between the plasma membrane and the wall, so they have an inside-outside within themselves there. I've got the overall formula for cellular respiration across the bottom here. See if I can get it all in view. Yes, I can. Glucose, C6H12O6, plus 6 oxygen will yield 6 carbon dioxide, 6 waters, and 36 ATP. So very handy to have everything as sixes again 6 carbons, 6 oxygens, 6 CO2, 6 water, and 6 times 6 is 36. I say a net yield of ATP because you're going to see in a moment we're going to end up needing some to get started, so we're going to burn some to begin with. If you want to know where they're all coming from, 2 of the 36 are coming from glycolysis, 2 are coming from Krebs, and the electron transport chain in the end will yield 32. So this is way more energy than photosynthesis is getting for a plant. This is the way that organisms are getting energy to use for functions, reactions, building things, breaking things. This is the business. Okay, so this I'm gonna now move away and go to our graphic organizer. Let's see if I can zoom out. There we go. It's about as much as, here's my old one. Got a desk of pens here going on, of course. We're gonna start here, work our way down, go across. This bubble you can ignore. It's a remnant from something that you don't need to know anymore, and I forgot to take it off. Then we're gonna go around here, and then finally end up back here. There are zoomed in photographs of each of these posted in the, like I said, PowerPoint section. So there's a picture of the whole thing that you can zoom into, or you can look at specific parts of it just one by one. Down here is fermentation, so we're going to talk about that last. You can kind of leave this stuff off to the side here for now. I've also got Leo the lion goes grr as a reminder here. Leo, I'm using red arrows for the molecules where they're losing their electrons. Grr for gaining electrons reduced. Where do those electrons end up? So I've got those kind of arrows all throughout, but I'm not going to specifically talk about it each time. We've covered it in photosynthesis. In the end, we are oxidizing glucose and other biological molecules like proteins or fats, and we're breaking them apart and getting those electrons over here to the ETC. Enjoy the Disneyland of the mitochondria, basically. Okay, so we're going to start with glycolysis. Glycolysis, glucose breaking. Glycolysis, I've got a couple of the steps here. All of these are going to be carbons, these little gray circles of mine. I just didn't write carbon C's in all of them. The formula, C6H12O6, and if you wanted to draw a glucose molecule, in the past we've also been drawing them as just hexagons, but I put a couple more details on here for you. So in reality, you'll see in your textbook, or if you go on to be a doctor or nurse or anything like that, glycolysis is actually 10 separate steps. There's a lot going on. There's a lot of enzymes involved. You do not need to know all those details for the purposes of AP Bio, but you do need to know that this is happening in the cytoplasm, right? The end result here is going to get moved into our giant mitochondria over here. So know that glycolysis is happening just in the soup of the cell, in the cytoplasm, and there's a lot of enzyme steps to make it occur. We're going to break glucose as a 6-carbon molecule into pyruvate as 2 independent 3-carbon molecules, and we'll get 2 of them as a result. What are we gaining and losing in the process? Well, in order to do some of the first initial breakings and adjustments, we're actually putting phosphates on glucose, opening up the ring, making it straight. We're going to need to spend 2 ATP to do that. So we actually are consuming energy in the very first step of cellular respiration, but we're going to get a big payoff in the end, so it doesn't matter. So we've got 2 ATP getting spent, so TP triphosphate to DP diphosphate, we're losing phosphates. Hey, why don't we just put them on either side of that molecule? We're going to do some breaking, we're going to put more phosphates on, and then we're going to start taking electrons off. So we have a new electron carrier. I've got them here. Unlike in photosynthesis, NADP, and changing to NADPH, we've just got NAD plus changing to NADH. So there's no P on this electron carrier. You can remember the P is the one for photosynthesis, but it's bringing its friend FADH2. Do not need to know their full names. They are just molecules that basically will take excited electrons off, carry them temporarily, and in the end they're going to be these shuttle trams that move them all to the electron transport chain where they are released. So each of these molecules, because there's two of them, they're going to offload electrons. So we're going to oxidize the molecule. I call them like poops. That's why there's a little mini poop emoji here. No, they're not actually. You don't even know their name, but just something fun. And the electron carrier is going to get reduced. So I drew in some electrons on the electron carriers there, and they're going to basically like end up going that way. Towards the latter steps of glycolysis, we're actually going to be able to generate 2 ATP from whatever chemical change happens here. It looks like removing the phosphates from the 3 carbon molecules. And each one is going to generate 2 ATP because of that. So we've gained 4, but we've spent 2. So that's why glycolysis has a net gain of 2 ATP. Our end product is pyruvate. That is a name you should know. It does show up on the standards. It's kind of the intermediate step. We've broken down glucose, but we're not yet releasing carbon dioxide. So to pyruvate form, we're going to follow the journey of one of them. So over here in the Krebs cycle, all of this is occurring for each of the pyruvates. So each one is going to go through, so all these numbers you can basically double for anything happening in the Krebs cycle. Krebs cycle, named by somebody named Krebs, has another name called the citric acid cycle. And down here I kind of like did some of the location pieces. So we've got our outer membrane, our inner membrane, the intermembrane space. This Krebs cycle is detailed and it's happening within the matrix. So that's what I've got. Another name for the Krebs cycle is citric acid cycle. Unfortunately, Calvin cycle for photosynthesis, all about the carbons. This is also all about the carbons. I personally struggle with Krebs, Calvin, trying to remember who's in which process. This has less C's. I don't know, like find a little trick or a little post-it on your bathroom mirror to help remember those for this upcoming unit. So a couple of things I've added. I did some of the carbon atom counting like we had on our photosynthesis graphic organizer, and so that's kind of an addition that I put on here. Again, this bubble you can ignore, but we're going to follow the pyruvate. So it's a 3 carbon molecule. This is a transition step before it gets into the mitochondria. All of this happened in the cytoplasm, and we're actually going to be releasing 1 carbon dioxide initially in this transition step. So we're going to take a 3 carbon molecule, break it down smaller into a 2 carbon molecule, and that 2 carbon molecule is going to move its way into the mitochondria. So you can imagine that outer surface of the mitochondria has a whole bunch of transport proteins that are designed for letting that 2 carbon molecule inside. The CO2 gas is just going to diffuse out of the cell through the cell membrane, get into the blood, into the lungs, you breathe it out. Here is one specific enzyme, coenzyme A. If you want to write coenzyme A, I may as well do that. Give me a second. This is kind of like Rubisco in photosynthesis's Calvin cycle. It's not an enzyme you specifically need to memorize coming into the AP exam, but this is something that they might pose a question about. They'll give you information, they'll tell you about it, at least here you've heard of it. So what I've drawn here is the 2 carbons coming from pyruvate leftovers, and I drew those 2 here, but with this spiky green enzyme attached to it, representing coenzyme A. So this molecule now is acetyl-CoA. That's a name that I keep seeing in diagrams, not one you need to memorize for the final this year, but you may see it on the AP test, again, presented with its information, not that you have to know everything about it. But very much so here we've got 2 carbons with the help of coenzyme A, so this acetyl-CoA complex is going to come and bond with these 4 carbon atoms from this leftover molecule coming around the bend. So 4 carbons plus 2 carbons is 6 carbons, and that is citrate. This is where the name citric acid cycle comes from. So 6 carbon atoms, take it down to 5, means we had to lose some carbon dioxide and get rid of one of the carbons. We do it again, 5 to 4, lose another carbon dioxide. In those processes, the electrons that were helping to hold on those carbons and oxygens, they get donated and put onto the electron carriers. Again, electron carriers carry the excited electrons, and they're going to offload at the ETC. But that's all now happening within the mitochondria itself. So we've lost 1, 2, 3 carbon dioxides. These are now just regenerating steps. So we've got 4, we have 4 again, 4 again. Hey, look, another incoming 2, lose 2, 4, 4, 4, add the 2 up to 6, 5, 4, 4, 4, etc. You will not need to have memorized the total, like how many does the Krebs cycle offer of the NADHs and the ATPs. You will just need to know the overall numbers. There's a new electron carrier. So NADH is kind of new, slightly different from the one from photosynthesis, but FAD, getting 2 hydrogens added, is pulling electrons as well. So this 4 carbon molecule loses electrons, and FAD gains them to become FAD2. So we're cycling around. Alternatively, in photosynthesis, we were trying to fix the carbon, bring the carbon dioxide in and get it incorporated into biological organic molecules. But here we're taking these, what we call intermediate organics, and we're taking carbons off of them. So we're losing the carbon dioxide. So it's the opposite process. Let's see what else I have. Yeah, I have that summarized here. Krebs cycle, releasing CO2 from organic intermediates. So when we say organic, we just mean things that have carbon to carbon, carbon-hydrogen bonds. So this again is occurring in the matrix. So it would be happening here in the middle. If you want to draw a little, Ferris wheel of Krebs cycle, go for it. Okay. So our excited electron carriers are loaded up with these excited electrons, and what I have written there is electron carriers take excited electrons to the ETC. So that's what all these dotted lines, they're going this way. I've just colored my ETC various shades of green. You could pick any color you want, it's not important. So ETC, again, for electron transport chain, very handy, same process. And we also have our old friend ATP synthase. This is the one enzyme you do need to know in the end of all of this. So I'll talk about these extra things on the outside in just a moment. Our electron carriers are coming in, and they're going to lose those excited electrons, and in that case they're going to go back to their oxidized form. This is their reduced form carrying their electrons. They're going to lose them to the proteins of the ETC over here. By doing that, we have these excited electrons. They're going to have the energy capable of pumping protons from the matrix to the intermembrane space. So I've drawn up arrows. They're not leaving the mitochondria. They're just going from the matrix space to the intermembrane space in between. So all these excited electrons getting offloaded, they're kind of going from that high energy state because they were already in organic compounds that are high energy bonds, and they're basically like falling down in energy, and with that release of energy we can pump protons. Probably sounds really familiar. We've been talking about gradients a lot. So we have an increasing proton gradient. The only way to release it is through ATP synthase. Oh, and I'm realizing I forgot. The only way to make ATP is from ADP and inorganic phosphates. You could put 32. I don't see how you're going to fit that. That's okay. So ATP synthase, again, a transmembrane protein, but it's on that inner mitochondrial membrane. So it's allowing this high gradient of protons to flow down their concentration gradient, and those protons are going to end up being part of water. You don't need to worry about where all the protons are coming from because they're coming and going, getting broken off of glucoses and kind of they're remnants of all these oxidation reactions along the way. All of these molecules are losing electrons. These are losing electrons, so there's a lot of protons available in this organelle. This is a word that's new. It may have already been on some things that you've seen, but I'm actually defining it here. So chemiosmosis sounds like chemical osmosis. We know that osmosis is the movement of water down its concentration gradient from where there's lots of water to where there's very little water. Well, this is the movement of ions down their electrochemical gradient. So protons from a high area of protons moving to where there's less protons. The only way to do that is through ATP synthase. So you could say that ATP synthase is powered by chemiosmosis. So we've made a whole bunch of ATP, 32, and that is the 32 from the electron transport chain, plus 1 for each pyruvate going around, so that's 32 plus 2, and the net 2 here, so that's where I'm getting the 36 overall. Other things, oxygen gas, right? They always say that you need oxygen to do cellular respiration. It's because oxygen is the final electron acceptor. This is the gas that is willing to take these now lower energy electrons. They were high energy electrons doing organic bonds, but they've kind of spent their energy in the ETC to make the gradient, to make the ATP. And now where can they go as lower energy electrons? Are inorganics, right? Oxygen's going to accept them with these protons, and we're going to end up with water. So this will probably be leaving as water vapor as an organism breathes out. This is aerobic respiration. Aero is with oxygen. So the electron transport chain can only function if there is oxygen available. Otherwise, you're going to start to get a dead end and a backup, and things can't offload because the electrons have nowhere to go. So things will come to a grinding halt. Also, if any of these proteins are not working. One special name of one, wondering where I can fit it, I'm going to try to squeeze it in here somewhere, is cytochrome C. I'm going to squeeze it under here in ATP synthase. And just draw an arrow somewhere. If you wanted to know which one specifically it is, you could look it up in your textbook. But one of these proteins within the electron transport chain is cytochrome C. It has a very highly conserved structure across lots of species throughout our animal kingdom. Because if you screw up this process, you're not getting ATP and you're not alive. So evolutionarily, this hasn't really changed much, and the structure of cytochrome C is fairly consistent, and we can track changes in it to look at evolutionary relationships of species. So you might see cytochrome C get mentioned. If you break it, you're dead. That it doesn't work after that. So we also have heat energy getting released. Anything that's alive has incoming food. If it is a warm-blooded animal or an endotherm having to make its own heat, a lot of these oxidative reactions kind of all over here are tearing and breaking bonds, and so you can be releasing heat energy. You're not going to get it all here in ATP. It's not a all energy conserved process. You're going to lose some, and so that energy, heat energy, can be released to help regulate temperature. If you're not eating, you're probably cold, right? If you're not eating, you're going to end up dead because you don't have this either. So, and the last couple parts here, we've got oxidative phosphorylation. So we talked about photophosphorylation in photosynthesis, where it's the original source of energy was light energy that helps us make the ATP. Well, here, now we are actually doing redox reactions. We are tearing electrons off of things and using those electrons in an ETC. So we did oxidization of biological molecules. I'm saying biological molecules because it's not necessarily just glucose. You could be breaking down proteins. You could be breaking down fatty acids from fats in your diet. And so you can still get energy from other things besides glucose. There's just a few more steps to get it to this stage to be moving into the Krebs cycle. So I've got oxidative phosphorylation. We've got biological molecules with excited electrons. We remove them. We put them on an electron carrier, NADH, and the final destination is to the ETC to form ATP. And in the end, oxygen's going to pick them up and we're going to form water. So it sounds really similar to photosynthesis again. It is basically the reverse reaction. If you flip it around, you're going to get photosynthesis. So instead of a donor, here oxygens with protons are going to take those electrons as acceptors and form water. I wouldn't say that water is the final acceptor because we're not adding it to water. We add it to oxygen to then form water. Some mathiness down here. We love some carbon math. So glycolysis, taking 6 carbon molecules, turning it into 3 carbon molecules. Each pyruvate ends up releasing 3 carbon dioxide. So we've got 1, we've got 2, we've got 3, but we have each glucose makes 2 of those, so that's where we're getting the glucose ends up releasing 6 carbon dioxide. So C6H12O6, all 6 of those carbon that were incorporated in high energy bonds as an organic molecule end up being released as 6 independent carbons in inorganic molecules, so low energy, and they're going to get breathed out to the atmosphere. All of this happens if you have enough oxygen. If you don't, we're going to do fermentation. So there is a term in your book, but my phone is resting on the book right now and I'm forgetting. Oh yes, obligate no, I'm wrong. We'll talk about it tomorrow because I don't want to make the mistake here. But fermentation is something your body's going to do if we cannot go that way, right? If we don't have the oxygen available to accept the electrons and this is grinding to a halt, well then we still need to keep this going because you can get some ATP. A net of 2, which is little to none, but it's something, right? And you have incoming food. So like, are you sprinting, right? Are you holding your breath underwater and diving? Like any of these reactions where the oxygen conditions become too low to be able to continue to do aerobic respiration over here. We then call that anaerobic respiration. So I've got that at the bottom. Anaerobic is without oxygen. This is lactic acid fermentation is this example that happens in humans. Alcohol fermentation is another example that yeast will do. This is how we brew beer and wine. I don't think yeast makes the wine, but the bacteria that we seal in airtight barrels don't have oxygen anymore after a certain part, so they will begin producing the alcohol. They will only get to a certain point before it gets too toxic for themselves. So that's why alcoholic drinks will only reach a certain proportion of alcohol in them. So this pyruvate, if we can't continue, it's going to get transitioned into lactic acid. You might also see lactate. I write feel the burn right here because we've got electron carriers coming in, losing their electrons and becoming oxidized, and then the lactic acid has gained and become reduced. So it's got a couple more bonds here. It's rearranged things. You do not need to know the unique structure of these. We haven't added or removed any carbons, but I say feel the burn because let's say you're sprinting. You can only breathe so quickly, but you're asking your muscles to move and spend ATP at a rate much faster than you can regenerate it. And so you begin to like run out of oxygen because you've sort of depleted this process. And that's when your body begins building up lactic acid. It's gonna cause you to have cramps. It's gonna cause that like soreness of muscles the next day. But this can be mixed up with things like when you work out really hard and tear a lot of muscle fibers, that can also be a cause of sore muscles. So this is not the only reason, but it does make like your muscles cramp and things like that. So I have written on the side here, if I can read it, because I'm looking behind my phone. Right. Fermentation needed to regenerate electron carriers, so at least you can keep doing glycolysis. So I've got this now oxidized electron carrier, not carrying any electrons, moving up here, and it can go back and help glycolysis to keep finishing. So this will still cycle between glycolysis and lactic acid fermentation if you have really low oxygen conditions. I think that's kind of it, folks. So we will be able to cover this in class on Thursday, tomorrow, January 16th. Please come with it filled out. You can ask questions. We can go over confusing parts. I can do some dances, but there is no chlorophyll going on. It's just a whole bunch of ATP synthase and ETC. This is the Disneyland of the mitochondria, and these are your shuttles taking excited electrons there. So we will have a few more details to work out before the end of the unit, a couple more details about fermentation and how organisms will use their fitness, like how are some adapted to doing this really well or others not so well. How does it change for small animals like a mouse, big animals like an elephant, polar bears versus hummingbirds, and we'll kind of do some compare and contrast. So please come with it completed. You can put this in your lab notebook or on the back of your graphic organizer, you decide. And again, all photos of this are posted in the PowerPoint folder of Schoology, and we're still in Unit 4. We're in enzymes, photosynthesis, and this is the last part of cellular respiration. So you can say goodbye to my kitty. There's Jackson hanging out right by the heater. That's basically where he is all the time now. So signing off, and have a great Wednesday night. Hopefully we'll see you tomorrow. Bye-bye.